where we left off, we had talked about DNA, its structure, how it stores information. Then we talked a little bit about RNA and how RNA is different from DNA. So what we're going to talk about in the last section of this unit is sort of how DNA and RNA work together to actually guide protein synthesis. So like we said, the genetic code that's stored in our DNA is determined by the bases. It's determined by the sequence of those nitrogenous bases. Those A's, T's, G's, and C's, the order that they are arranged in is what stores information. Just like arranging the letters of the alphabet can form different words, arranging these nitrogen bases in different ways can form different proteins. There's about 3 billion base pairs in the human genome, is what we call it, all of the genes that an individual has. Okay? And those base pairs, actually a large proportion of them are in non-coding regions of our DNA. So they don't actually have instructions for building protein. And some of those are regulatory, they tell when to turn genes on or off, and some have no apparent function. But if we sort of look at the basics of how this works, we'll go through it just simply and then talk about some more of the details. First thing is that the DNA code, which we know is in the nucleus, in the chromosomes, stays in the nucleus. And when a section of DNA, when a protein needs to be made, the instructions for how to build that protein are copied. They're copied from the DNA into an RNA molecule. It's, the type of RNA is called messenger RNA. It's the first step. The gene that tells how to build the protein gets copied to messenger RNA. Because messenger RNA can leave the nucleus. Okay? It's a smaller molecule. Leaves the nucleus, goes out into the cytoplasm to the ribosome. Remember when we talked about cells? The ribosome is where proteins are synthesized. So it's bringing the instructions to the ribosome, which is going to start putting together this protein. Once the messenger RNA is at the ribosome, okay, another type of RNA, transfer RNA, starts bringing in amino acids. Protein's made of a series of amino acids. And so the messenger RNA has a code for which ones should be brought in. The transfer RNA brings in the correct amino acid. Then another transfer RNA brings in the next amino acid. And so on and so on. Stringing together these amino acids, kind of like putting pearls on a necklace. Okay? And it starts to become longer and longer, this chain of amino acids. Okay? They're put together in the correct sequence. And once all of the amino acids have been attached and the protein is finished, it detaches from the ribosome. We know that it folds up into its complex structure that allows it to work, and the protein is synthesized, it's made. And that's how protein synthesis works. It's how the DNA and RNA work together. There's a couple terms for protein synthesis when we look at the details. They're called transcription and translation. Transcription refers to taking the DNA code and copying it to messenger RNA. So this works through the action of various enzymes. Okay? And so here we obviously have a strand of DNA. And if we imagine a section of that DNA, so one strand of DNA that's in a chromosome has thousands and thousands of genes on it. If only one of those genes is going to be used to make a protein, what happens is enzymes sort of unzip a section of the DNA so that it's exposed, that the bases are exposed, they're no longer bonded to each other. Okay? And then an enzyme called RNA polymerase. It's an enzyme. It's making a polymer, RNA, based on the DNA template. So this enzyme, okay, forms a strand of messenger RNA that's complementary to the DNA bases. For example, if in the DNA base, if the first base is a G in the DNA, what base would there be in the RNA? C, and so forth. How about here?
T.
No.
O.
Not O.
U.
U.
Oh yeah, because yes, uracil.
Yes. So the difference is that RNA doesn't have a thymine. It has uracils in place.
So why is it that A ends up with a T?
Because it has adenine, so they're still complementary, but it doesn't have any thymine in it. The U replaces the T in the RNA strand. So it has C's, it has G's, it has A's.
Yeah, but wait, there's
T.
T and T.
That's what I was asking.
Where?
There's
The
T.
A and
Oh,
the-
yeah, that should be you. You're right.
Oh.
No, that should be you. Sorry. And so this strand of messenger RNA now contains complementary base sequence that's based on the DNA, and it can then carry those instructions out. So this process is called transcription, taking DNA, copying it into messenger RNA. The part when that messenger RNA is then used to create a protein is called translation. In translation, the code is read, and the protein is actually synthesized. So the way the code works is that every 3 messenger RNA bases is a code for 1 amino acid. Okay? Each 3 bases is called a codon. So here we have our strand of messenger RNA. Every 3 bases is 1 codon that has the code for a single amino acid. Okay. The messenger RNA, it binds to the ribosome. And then another type of RNA, transfer RNA, starts bringing in the proper amino acids. The way this works, a transfer RNA molecule has these loops. Kind of looks like a clover leaf. And down here on the anticodon loop are some bases. They're called the anticodon. So if the codon on the messenger RNA is GAG, the anticodon is complementary to it. So it would be CUC. And then, it's not shown here, but attached to this region of the transfer RNA is the amino acid. So GAG is a code. The genetic code, that is a code for 1 amino acid. I'll show you how you figure out which amino acid in a minute. Okay? And so it brings in the proper amino acid. Then another transfer RNA would read the next codon, CAG. What would the anticodon for CAG be? C, GUC. Okay, so it matches up. It brings in a different amino acid. The amino acids get bonded together, and now the protein starts being made, 1 amino acid at a time. There are special codons called start and stop codons. The start codon initiates the beginning of the protein. When the stop codon is reached, that tells the ribosome that this protein is finished. All the correct amino acids have been strung together. it's done, and it can then be released from the ribosome to do whatever it needs to do. This is the messenger RNA codon chart. This tells you for any mRNA codon what the appropriate amino acid is. So if a messenger RNA codon was AAA, the way we use this chart, and it can be set up lots of different ways. Bless you. We take the first base. We start on the left side. So if the first base is an A, okay, there's A. Then we look for the second base. Let's say it's an A as well. Here's A on the second base. And then the third base, A.
Thank you.
If we look where all three of those intersect, we see AAA is a codon for lysine. That's always. So whenever the messenger RNA has AAA, that's a codon to bring in lysine as its amino acid. How about if we look up UCG?
Sir.
Yeah, these are three-letter abbreviations. Serine is the amino acid that bring in. Okay. GAG.
Blue.
Glue. Okay. GAG. Okay. You'll see. How about UGA?
Stop.
Yeah, that's the stop codon. That's the one that initiates the completion of the protein. Okay. Start is over here.
Where metin started just like the same.
Yeah, methionine is a special amino acid that indicates the start of the protein. And there are some repeats, for example, things, UCC and UCG are both codons for serine. You have some overlap there, but this is the universal genetic code chart. And it really works for all organisms, whether it's a bacteria or a mushroom or a human or a pine tree. The code is universal. In all those different organisms, one genetic code is a symbol for one amino acid.
Well, we have to memorize that for the test.
No, you always have that available. You have to know how to use it. So when we look at just sort of the overview, inside of the nucleus, we have our DNA strand, which is used to synthesize a complementary messenger RNA strand. This messenger RNA then leaves the nucleus, goes out to the ribosome. What is this called, this process? Transcription, and this is translation. So this messenger RNA moves out to a ribosome. Okay. Each, how many bases? 3 bases is one codon. These transfer RNA molecules bring in, you can see, here's the protein chain that's being assembled. This transfer RNA molecule is bringing in the next amino acid in sequence. This one would bring the next one. And just continue stacking them together, bonding them together until the protein is finished. So, the one gene, one polypeptide model is used to sort of explain how our DNA works, how the DNA code works. I mean, it's basically the concept that for each gene, a gene is a code for one protein, how to build a single protein. Okay? But when scientists look at genes, and scientists are able to use modern biological tools to sequence DNA very quickly nowadays. That they can take a sample of cells and basically a sample of DNA, amplify it so there's a lot of the DNA, and then use computers to read the sequence of bases. This used to take a very, very long time. It used to be very complex. But it's become much, much easier with the advent of modern technology. But scientists have learned that there are huge regions of our DNA. They used to be called junk DNA because they thought they didn't do anything. They're these long sequence of often repeating letters that didn't seem to code for anything. Okay? It's actually up to 90% of the DNA is this non-coding region. But we now know today that some of those non-coding regions do have functions, regulatory functions to cause other genes to turn on or to turn off. So these are, they are important parts of the DNA. Okay? And what happens is the coding regions are called exons, okay? And this DNA gets sort of spliced together to form the complete gene. A gene could have interruptions of non-coding DNA in it that doesn't actually code for anything. Okay? All right, any questions about DNA?
Are we getting the packet?
Yeah, I'll give it to you right now.
Wait.
Wait, what packet?
Yeah.